Hi, this is the presentation for the part of Chapter 24 that we're going to do. Chapter 4 is medical genetics, and we're doing only sections 3 through 6. I don't have the entire chapter cover here, so that's all you're responsible for are those sections. And the first part of this chapter just talks about the different types of genetic disorders like autosomal recessive, autosomal dominant. And we kind of talked about those already in other contexts in the class when we did Mendelian genetics. So this is talking more about evaluating and treating genetic abnormalities. So genetic testing is using a test to discover if a particular individual has a particular genetic abnormality. Whereas genetic screening is screening a population to see the prevalence of a particular genetic abnormality. So one is done on the individual basis, one is done more on a population basis. And so there are a number of different methods that are used, ranging from biochemical assays from like a blood sample to actually doing a gene analysis. Excuse me. So there are some techniques that are available that can actually detect a genetic abnormality without actually doing a test for that particular gene. And these are primarily disorders that affect proteins, often enzymes. And so there are biochemical methods used to detect for a functioning enzyme. And so you don't actually even need to do a gene analysis, but you can check the, the measurement of enzyme activity. Tay-Sachs disease, which we watched a video about earlier in the semester, the neurological disorder, it's autosomal recessive. So that can be tested for in a blood sample through enzymatic assay. The enzyme that is defective in Tay-Sachs disease is HexA. And HexA is involved in converting glycolipids in the brain to N-acetylglucosamine. And the assay uses a fluorescent tag on a kind of artificial substrate for the enzyme. And so if the enzyme is present, it converts that substrate to a fluorescent molecule. The enzyme isn't present, then you do not get fluorescence. So it's a pretty easy detectable mechanism. And so homozygotes or normal that have two functional alleles will show a high level of fluorescence. Individuals with Tay-Sachs disorder will have little or no fluorescence, and then heterozygotes have an intermediate level. So that's a way of kind of determining whether you're homozygous dominant, heterozygous, or recessive by doing an enzyme assay linked to the particular disorder, the enzyme that is affected by that disorder. And there are a number of these that are available using various biochemical detects. But many other disorders are detected at the DNA level where they actually look for the abnormal allele to determine if a disorder is present or not. And that can be done by various, some by electrophoresis. It kind of depends on the particular disorder. Another type of testing that's done to detect abnormalities in chromosome number is doing a karyotype analysis. And so a karyotype is where you do analysis for the chromosomes and you look for abnormalities in number of chromosome or size of the chromosomes, either being short or too long. And most abnormalities in chromosome number do not result in live birth. So it's one of the primary causes of spontaneous abortions during pregnancy is chromosomal abnormalities. And then some of those that do survive to birth die in infancy. So there's only a few chromosomal abnormalities that are survivable. And again, during a karyotype of women or of a blood sample or of fetal cells can detect the chromosomal abnormalities. And these increase with maternal age, and so they're more commonly recommended in pregnant women over 35 years of age to see if they have chromosomal abnormalities. There are also a number of other genetic disorders, and we've talked about this a little bit already, that are screened for at birth in newborns, such as PKU. We've talked about this, where usually a heel prick or some small blood sample is taken at newborns, and that is another one of those enzyme assays that they can use to detect for a variety of genetic disorders. There's lots of issues with doing genetic testing because we now have the ability to do genome analysis, and so they can test to see if you have a whole number of genetic abnormalities. And so there are both ethical issues and personal issues. We talked about the video we watched about Huntington's disease, and they said that often people don't want to know, even though they can get a test done to see if they have the Huntington mutation. They prefer not to know because there's no treatment, and whereas other individuals will like to know. There's issues with like insurance companies of what information might be available for like employers or insurance companies to discriminate against either hiring or insuring someone because of information on their genetic test. Okay, so genetic testing that can be done prior to birth on fetal cells, the most common one is an amniocentesis. And an amniocentesis is done with a syringe that is used to remove a sample of amniotic fluid, which would contain fetal cells. And then those fetal cells can be, a karyotype can be done on the fetal cells to look for chromosomal abnormalities. That the problem with it, you can't generally use maternal sample like a maternal blood sample because intact fetal cells do not pass into maternal blood. So the only way to actually get fetal cells is either through an amniocentesis. There's another mechanism where the chorion, which is part of the placenta, which is actually derived from fetal cells rather than maternal cells. Some of the placenta is maternal origin. And you can do a scraping and remove some of those cells and do a karyotype on those cells. Both of these are not without risk. They're invasive. There is, particularly with the chorionic villus sample, and that's why it's not done as much, there is an increased risk of pregnancy, miscarriage. There are some methods developed to look for some of these abnormalities. And for example, there is a blood test now, maternal blood test for Down syndrome that looks for pieces of fetal DNA in maternal samples, and through DNA technology are able to kind of put those together and see if there's too much of the DNA, for example, from chromosome 21, which would be suggestive that the fetus has Down syndrome. So there are some advances being done in trying to get less invasive procedures for detecting chromosome abnormalities prior to birth. You do have the possibility, and I have a video about this, which I might show you when we talk about genetic forms of cancer, but another technique that's used is in conjunction with other genetic testing is in vitro fertilization, where sperm and egg are combined outside of the mother's body. And then when the fertilized egg has divided a few times and is at the 8-cell stage, you can actually remove 1 or 2 cells from the embryo at that stage, and it is not harmful to the embryo. And so you can remove 1 or 2 cells and actually screen those cells for genetic defects and determine whether or not that particular embryo has a particular genetic defect. So that would be done where an individual knows they have, like say you have the Huntington's mutation, and you could screen embryos to see if you have a 50% chance of passing that mutation on. So you could screen embryos to determine if you have one that does not have the mutation, and then that could be implanted back into the mother and carried through pregnancy. And again, there's obvious ethical questions with this procedure also, but it's another technique that's available for in conjunction with genetic testing. For some reason, your book now goes to a completely different topic, which is prions, which has really nothing to do with genetic testing. But anyway, it's an interesting. Prions are proteins, and they're sometimes called infectious proteins. They're not really infectious in the traditional sense of infections, like where you get a bacteria and that bacteria goes on to divide and produce more bacterial cells. Excuse me. Or a virus goes on and produces more virus particles inside of your cells, and then you can pass that on to individuals. But prions are these abnormal proteins that can cause various neurodegenerative diseases. It's kind of the same disorder. It's called different things in different organisms. In cows, for example, and it's called mad cow disease. In humans, it's called Creutzfeldt-Jakob disease, which is the same as mad cow, and it's due to a prion. And prions are a protein that can exist in 2 different conformations. And the normal one does not cause the disease. The abnormal form can cause, let's say, mad cow disease. And and certain cells have low levels of this prion protein. But what happens is if you have a cell exposed to this abnormal prion protein, a normal cell, it can cause the spontaneous conversion of normal proteins to the abnormal proteins. So that's why it's kind of like an infection, because if you ate meat, say from a cow that had mad cow disease, and you would have to eat neural tissue because it's only found in neural tissue. So clearly, like if you were eating brain, But there's neural tissue, like in muscle, you have nerve fibers running through muscle. So hamburger and things may have some neural tissue in. So if you ate contaminated meat where they had these abnormal proteins and they got into your neural tissue, it could spontaneously cause the formation of these abnormal proteins in your tissue, and that's how you would get Jacob-Creutzfeldt disease. So there have been various outbreaks of this, and there's lots of work done to kind of register cows and sheep. It's also been found in sheep. And so that if there is an outbreak, it's easy to trace what other animals might have been exposed. It also used to, and sometimes be due to the practice of taking kind of leftover pieces from slaughterhouses and grinding them up and incorporating them into cattle food as a protein enrichment. And so if there was an infected animal, you're now feeding it to many other animals. There was an outbreak a number of years ago in England, and so the US would not allow any beef from England to be imported into this country. That is no longer true, but that's how it's kind of considered to be an infectious disease, but it's not really a genetic disorder in kind of the classical sense, except that it is an abnormal protein that causes it. Okay, I think I already went through that of the abnormal proteins in the brain. Okay. So that did not really seem to fit into anything, but it's one of the sections in this chapter. Gene therapy, and we've talked about this a little bit already, is the idea of being able to cure genetic disorders by getting the normal gene introduced into living cells. And we talked about this where there's been a recent case of success with this, with sickle cell anemia. And in theory, this could also, that's my grandfather clock again, chiming. Besides being used to treat genetic disorders, it could potentially be used to treat things like cancer, heart disease, maybe combat infections such as HIV. But it's still, success has been very limited to date. But it is an area where active research is being done because the potential of gene therapy is kind of endless. And I mentioned this, I think we talked about this a little bit last time, so I'm not going to spend a lot of time, that the problem with after a person is an adult or has been born even, it doesn't have to be an adult, you now have millions of cells, and if you're trying to correct a defective cell, you need a way of getting the gene, the correct gene, into the human cells to replace the defective one. And the two methods that are primarily used is either a viral vector, in which the gene that you want is incorporated into a deactivated virus particle. Usually retroviruses are used because they can get incorporated into your DNA through the reverse transcriptase, and it would be deactivated so you wouldn't actually get sick from whatever virus particle that is. And so that would be a way of getting the correct or the corrected gene into the cell's genome. It's much more complicated than that because it has to be put in the right place. So the right promoters and things like that would be surrounding it to make sure that it would be turned on, turned off properly. The other one is the use of a liposome, which is kind of a little fake cell with just one lipid not a bilayer, but a lipid monolayer. And so you would have the DNA with the gene inside of that. And those can be taken up into cells. And so that would be a way of getting the gene into the cell and getting the DNA be able to be incorporated into the cell's genome. And so that's being looked at a lot with various genetic disorders as well as other diseases as a means of treating them. Another area that is a big growth area now is personalized medicine. And this has grown tremendously with the ability to do a genome analysis on an individual, and the cost of that, and we talked a little bit about this before, has markedly declined in recent years, so that you can have a genome analysis now, I think, for less than $2,000. So that the idea behind that is by using the genome analysis, you could get a profile of an individual's, for example, tumor. So you could do a molecular gene profile of a sample of a tumor from an individual that might have breast cancer or colon cancer. And because there are lots of different genetic abnormalities in different tumors, by looking at an individual's tumor, you might be able to target which genes are abnormal, whether it's more tumor suppressor genes or oncogenes, or what's being overexpressed. And that might enable you to personalize the drug regimen for treating a particular tumor that would make it hopefully work better on that individual based on the profile of their tumor. So that's a big area, and you'll even see different hospitals and stuff that will talk about personalized treatment plans for cancer, and that's what they're looking at, is doing the genomic analysis of your cancer. And this is just an example where they're doing one of a lymphoma and picking up which genes are expressed, which are red, which genes are not expressed, which are in green. And so this is one of two different individuals. And you can see that the profile of their tumors are very different. Even though they're both beta cell lymphomas, they have very different molecular profiles of which genes are being expressed and which genes are not being expressed. So that's the kind of information that could be used for individual treatment of various, particularly cancers. That's really similarly, just in general, with use of various drugs to treat disorders, individuals might have, because of their genome, have different rates of breaking down a drug or absorbing it from the digestive tract or excreting it or getting it into cells. And so by looking at an individual profile, you could help better individualize their treatment with various drugs based on their genetic profile. And that's called pharmacogenetics, where your actual drug regimen would be personalized based on your particular genome and how you would process a particular drug. And this is just an example. There's a drug called Coumadin, which is warfarin, which is it's what's called a blood thinner. And it actually is used for people that sometimes have high blood pressure or high rates of blood clots. So it kind of thins their blood, but makes it less likely to clot. And it's broken down by an enzyme in the liver, the P450 enzyme systems. And so different individuals have different activity levels of that enzyme. And so by knowing what an individual's level is, you would know the dosage of Coumadin that would be best prescribed. And because it is an anticoagulant, it's has to be carefully monitored, the use of Coumadin, because it can lead to abnormal bleeding because of its anticoagulant properties. So like, for example, if someone is on Coumadin and needs to have surgery, they have to, if it's a voluntary or scheduled surgery, they have to stop taking the Coumadin for a while beforehand, or otherwise the surgery is difficult because of they don't normally clot their blood. So that's another kind of use of personalized pharmacological doses based on your genome. So that's just more about the Coumadin. You don't really have to know anything specifically about Coumadin. I think that's the end of this section of chapter 24. So we will stop there. And if you have any questions about any of this material, you can just ask me.